Okay, welcome everyone. So this is Matt Bucker from University of Manitoba talking, and I've invited today's speaker, so I'm going to also give a brief introduction. So Bob Proctor is a scientist with the United States Department of Agriculture, and at the same location where I worked before I came to the University of Manitoba. So we were colleagues in the past, and that's how I got to know Bob. So Bob's also a Canadian, which I thought might make him willing to accept this invitation in part. So he's from BC and has his bachelor's degree from the University of Victoria. And then he went to University of Wisconsin-Madison in the United States for his master's and his doctorate in plant pathology. And I think, Bob, I'm right that you've had basically your entire career after that at the USDA. So that's kind of an unusual thing these days, especially. But Bob's got a great track record of really innovative and impactful research in genomics and evolution of plant pathogenic fungi, and particularly in the genus Fusarium, but also some other fungi as well. So I'm really thrilled to have Bob with us, and I think you'll all enjoy his presentation. So the format, we're going to have his seminar, and then we'll have just a brief break for washroom and refreshment, and then the fellows can return for a conversational meeting with the speaker after that. So I will turn it over to you, Bob.
Hey, thank you, Matt. I appreciate the opportunity to speak today. If you can't hear me at any time, just let me know and I'll speak up or try to change things. And before we got going here, I realized that I have to look at my presentation on the screen in front of me, so I'm not going to be looking at you, so please excuse that. But anyway, over the past decade, or really 2 decades, I've been involved in a combination of chemical, gene function, and genome sequence analyses that have revealed a lot about the genetic bases for mycotoxin production in fungi. And these studies have provided insight into the evolutionary history of mycotoxin production and why variation in production exists within and among species. And so that's what I'm going to relate to you today. But first, let's start with a working definition of mycotoxins, because there are a few. And so today I'm considering mycotoxins as toxic fungal metabolites that contribute to food, that contaminate food and feed, and that pose health hazards to humans and other animals. And one thing about mycotoxins is they're generally produced by fungi that are either strong or weak plant pathogens, and there's often or typically a correlation between disease severity and the level of contamination of mycotoxins. And so here are among 3 of the most important mycotoxin-producing fungi: Aspergillus, Flavus, Fusarium verticillioides, and Fusarium huminearum. And then so here's a slide showing the major groups of mycotoxins: aflatoxins, fumonisin, ocotoxins, trichothecenes, and zerelinols. So these are considered the most important mycotoxins, and they're regulated they're regulated in various countries, including the U.S. Not all of them are regulated in the U.S., but some of them are, and most of them are regulated in Europe. And as you can see here, they vary in structure. They tend to be produced by species of Aspergillus, Fusarium, in some cases Penicillium, and they affect a wide range of animals or livestock, humans, and dogs, so some pets as well. And they cause a variety of clinical effects, including in some cases liver cancer, reduced growth, and kidney disease, liver disease, and other clinical effects. And so these are the major mycotoxins that the USDA are most concerned about, and people in general focus on these ones. But mycotoxin-producing fungi tend to produce a lot of different secondary metabolites and A lot of them are considered mycotoxins, although in a lot of cases, minor mycotoxins. And their effect on human and animal health is not as clear as it is in the case of trichothecenes, aflatoxins, and fumonisins. So one of the when you consider, especially most of my work is on fusarium, and when you consider fusarium, species produce so many different secondary metabolites, including mycotoxins, and so over the past couple of decades, we've been looking at the genetic basis for variation in mycotoxin production. So what we see is multiple types of variation, and then we and others have found the genetic basis for those different types of variation. So here's 3 types of variation that we consider. That's the production of a mycotoxin versus non-production of the mycotoxin, and that results from the presence and absence of biosynthetic gene clusters. There's also production of different analogs of the same family of mycotoxins, and that results from variation in biosynthetic genes. And then there's also we've seen over the years, and then now we've really good evidence for it, is the truncation of biosynthetic pathways by non-random loss of genes. And so as I go through the rest of my talk, I'm going to be talking about these types of variation in mycotoxin production and how the genetic bases have arisen. And then I'm also, when you consider the variation in production and the variation in genetics, it raises the question, what factors drive variation in the genes and mycotoxin production? So I have some hypotheses about that I'll also be talking about as I go along. So we're all on the same page, I'm going to do a brief introduction to biosynthetic gene clusters. And so here is the biosynthetic gene cluster that confers the ability in fungi to produce fumonisins mycotoxins. And so the arrows represent genes and they point in the direction of transcription. And in this cluster, there's 16 genes, and they do a variety of things, but most of them encode enzymes that catalyze biosynthetic reactions that turn simple molecules into more complex molecules like fumonisins B1. So these, some of the genes encode enzymes. So FUM1, the FUM1 enzyme is a polyketide synthase that synthesizes this long polyketide. The FUM8 gene encodes aminotransferase that catalyzes condensation of the polyketide and an amino acid to form this compound, and then FUM6 catalyzes hydroxylation of this molecule, and then so on until we get a molecule like fumonisin B1. These gene clusters contain other genes that encode other kinds of proteins as well, including transcription factors that regulate expression of the cluster genes, and then also transporters that transport either intermediates within the cell and actually the final products, the mycotoxins, out of the cells in which they're synthesized. Mycotoxin biosynthetic gene clusters also can contain resistance gene that protect the fungi from the toxic effects of mycotoxins. So that's biosynthetic gene cluster and their function. And over the last 2 decades, I think, the biosynthetic gene clusters for most mycotoxins and a lot of other fungal secondary metabolites have been identified and characterized using various methods. And so here's examples of the gene clusters for fumonisins, ocotoxins, cerealinone, bovaricins, and amiactins, and then trichothecenes. And you can see they're just, some cases there are 3 genes, some cases, in the case of aflatoxins, it's a huge cluster. I can't remember how many, at least 20 genes. And then I think trichothecenes are notable in that they have, at least in fusarium, there are 3 trichothecene biosynthetic loci. There's a big cluster and then there's a 2-gene little cluster, and then there's a single gene cluster. So sometimes not all the genes for the biosynthesis of a particular toxin or secondary metabolite are present at the same locus, but typically they are. Okay, so now I'm going to talk about the distribution of selective selective mycotoxin biosynthetic gene clusters in Fusarium. And just to give you a little background on Fusarium, it's a species-rich genus that includes many plant pathogens and a lot of mycotoxin producers. And we've been doing a lot of genome sequencing of species and looking at so many species now that it's easier for me sometimes to use the species complex name. So species complexes are phylogenetically distinct lineages of Fusarium, and they're named after species that occur within them. So the Sambucinum, and I've abbreviated them here just using the species name. So the Fusarium Sambucinum species complex is abbreviated here as Sambucinum, and that's an important group of pathogens, and the Fusarium Fujikuroi species complex is abbreviated here as Fujikuroi. And so what we've done now is we've got a good phylogenetic understanding of the phylogenetic relationships of Fusarium species. Now we can use genome sequencing to map the presence and absence of biosynthetic gene clusters on this phylogenetic scaffold. And so here's an example of the presence and absence of 4 biosynthetic mycotoxin biosynthetic gene clusters: Fumasin, Fusarin, and Trichothecenes and Zearalenor. And so I think one of the first things that for me is obvious here is the variation in the distribution patterns of the different gene clusters. So Fumasin, this is not an exhaustive tree of all the species of Fusarium, so it doesn't show all species that have the Fumasin gene cluster, but you can see here that the Fumasin gene cluster, the distribution is somewhat sparse and it's discontinuous. It's in some species, but not others, and 2 closely related species can differ in the presence and absence of the Fumasin cluster. Whereas the Trichothecene cluster up here in the Sambucinum complex, at least, is continuous, but it has a much more limited distribution across the phylogenetic breadth of Fusarium. The Zearalenor cluster is similar, but even less distributed. And I included the Fusarin cluster here because it's widely distributed, but also it occurs sporadically among the species and species complexes. And I note this light, the light blue here indicates that within a species there can be variation in the presence and absence of a particular mycotoxin biosynthetic cluster. And this is particularly important in the case of oxysporum. Actually, I know of 2 strains of Fusarium oxysporum that have the Fumasin biosynthetic cluster, and hundreds of strains that don't have it. So, and in the case of Fusarium Fujikuroi, strains tend to have they have the Fumasin cluster, but sometimes the cluster is messed up, and so it's no longer functional. Okay, so when you see this variation in distribution patterns of various mycotoxins, for me the question was, how does it occur, and what are the genetic processes that lead to this variation? And so that's what I'm going to address here. And so let me go back. So the one thing is vertical inheritance. So the gene cluster occurs in an ancestor, and then as the descendant species diverge from that common ancestor, the species can inherit or not inherit that cluster. So the vertical inheritance seems to be what is important in the continuous distribution of the Trichothecene cluster in the Sambucinum clade and in the Incarnatum equiseti clade. In contrast, there can be gene loss, and what we're finding is that gene or cluster loss is rampant within Fusarium, and so this is a really good example here. That is the Fumasin cluster in the Fusarium Fujikuroi species complex. So this on the left here, you see a tree. It's a species tree that's probably, I can't remember exactly, but it's inferred from at least 10 or 20 housekeeping genes, which should give us an idea how the species are related to one another. And then when you see what we assume is that the humanism cluster was present in the common ancestor. And so anytime you see a black line, it means the cluster, we infer the cluster was present or is present, as it say in the case of Fusarium verticilioides here. But the blue line indicates that cluster is absent. So from this analysis, what we inferred was that there's been at least 20 loss events of the humanism cluster in the Fusarium fragiculus species complex. And so that would explain why the distribution of the humanism cluster is so sporadic in this species complex. So there's also another type of variation in presence and absence of gene clusters, and the black Aspergillus species, Aspergillus niger and Aspergillus welwitschia, and the presence and absence of the oak toxin cluster in these two species is a good example of this. So these fungi are plant associated, although they're not. They don't cause severe plant disease typically. They have this 5-gene ochratoxin biosynthetic gene cluster, and here's the structure of ochratoxin A. And what we find is that these two species exist as mixtures of producing and ochratoxin producing and non-producing isolates. So each species is a mixture of these two, a mixture of fungi with these two types of phenotype. So and that difference results from presence of two alleles. One allele is an intact ochratoxin cluster, and the other allele is a mostly deleted ochratoxin cluster with just a little piece of the OTA1 gene present, and then but the genes flanking the cluster, shown here in black, are also present. And so these two fungi have very similar alleles. And if you do a phylogenetic analysis of DNA sequences that are common to the two alleles, what you see is that the intact cluster in Aspergillus niger is more closely related to the intact cluster in A. welwitschia. When just looking at certain sequences, the intact cluster in A. niger is more similar to the intact cluster in welwitschia than it is to the sequences in niger with the lead cluster, and the same goes for welwitschia. So what seems to have happened here is that at some point in the common ancestor of these two species, there was an intact ochratoxin cluster which underwent deletion to create two alleles. And then these alleles were carried during speciation when niger and welwitschia split. And so the two alleles have been carried through a speciation event, and now both species exist as producing or as individuals that can either produce or not produce ochratoxin A. And we see this sort of thing a lot in some fungal species. So here are some of the other examples where species exist as producers and non-producers of various mycotoxins. So we see with aflatoxins, humanisins, in welwitschia, ochratoxin, and niger and welwitschia, and then humanisins in the species Fusarium fragiculus. And so this raises the question, what drives the occurrence of mycotoxin production versus non-production phenotypes within one species? And some recent work that I was not so recent, but I've become aware of it somewhat recently to explain this, and the work has been done in the Milgram lab at Cornell University, and it's on aflatoxin producing and non-producing strains of Aspergillus flavus. What they showed was that there is a fitness cost to aflatoxin production during interactions with microorganisms, interactions of Aspergillus flavus with microorganisms in the soil. So there's a cost to producing aflatoxins when interacting with those microorganisms. In contrast, aflatoxin production confers a fitness advantage during interactions with insects. So you can see there's competing cost benefit to production depending on where Aspergillus exists, and it exists both in contact with insects and in the soil. So there could be an advantage to have individuals that have either a producing or non-producing phenotype. So that provides an explanation for why fungi exist as with fungal species can exist as with two different phenotypes in terms of mycotoxin production. So that's covered loss, how production versus non-production can result from loss of gene clusters. There's also variation in production and non-production can exist by acquisition of genes, and I'm going to focus a little bit here on acquisition by horizontal gene transfer. And I came to this not knowing anything about molecular genetics or molecular phylogeny, and so when I do these analyses, I do a sort of a small battery of like 5 or 6 analyses to sometimes maybe too much, but I want to be sure that all the data that I see supports horizontal gene transfer. And so we do several kinds of analysis. And the first thing we see is branch conflicts between the species tree and mycotoxin biosynthetic gene trees. So we use bootstrap, constraint analysis, and reconciliation analyses to test whether those branch conflicts are real. And then I also do this synonymous site divergence analysis, which I'll describe in a minute. So I'm just going to go through these different kinds of analyses to show how they can provide evidence for or against horizontal gene transfer. So here's an example of what we thought was horizontal gene transfer of the fumonism cluster between species. And so what you see here is 2 trees. This tree on the left is a species tree inferred from multiple housekeeping genes. So it should show the relationship between the species. The tree on the right is a fumonism gene tree, so it shows the relationships between fumonism biosynthetic genes in the different species. And so on the tree on the left, all the species from Fusarium frugiculi down here, fourth from the bottom, up to the top, Fusarium coesis, are members of the frugiculi complex, whereas Oxysporum, Fusarium hostae, and the Huertae are members of different complexes, so they're more distantly related. And you can see from this, hopefully, that Fusarium anthophyllum and Fusarium hostae are relatively distantly related, given the species included in this tree. But their fumonism biosynthetic genes are closely related. Likewise, if you look at Fusarium bulbicola, which is a member of the frugiculi complex, and Fusarium oxysporum, the species are relatively distantly related, but their fumonism biosynthetic genes are closely related. And so this is a branch conflict between the species tree and the fumonism gene tree, and it suggests, when I see that, I think horizontal gene transfer. And so the first thing that can be done is look at the bootstrap values. So bootstrap, you just repeat the analysis, 1,000 or 10,000 times, and from this analysis, you see that 100% of the trees formed by those repeated analyses give you this branch. So it supports the distant relationship of bulbicola and oxysporum. And in this case, bootstrap value is 100. So every time you run the analysis, you get the same branch. So that supports the conflict. It says the conflict is probably real. We can also do a constraint analysis where I take the alignment data for the fumonism gene tree, and I ask a program, is the fumonism tree fit the data better than the species tree? And generally this constraint analysis agrees with the bootstrap analysis. So you can also do a reconciliation analysis, and I've been using this program called Notum. And what it does is it looks at a species tree and and then a biosynthetic gene tree, and tries to reconcile the branch conflicts using an algorithm, and it invokes either horizontal, when it sees a conflict, it invokes horizontal transfer, gene duplication, or lineage sorting. And my only issue with noting is that it seems to predict horizontal gene transfer too much. Regardless of that, so this is the output from a noting analysis. What you see is what I see when I do the analysis. But regardless, this noting analysis detected the horizontal transfer of humanism genes that I predicted between Fusarium hospi and Fusarium amphiphyllum, and between Fusarium bovicola and Fusarium oxysporum. So the last type of analysis I use is this synonymous site divergence. And what it uses is estimates of the number of synonymous changes per synonymous site. So it looks at codons and synonymous sites within codons, and as a measure of genetic divergence. And so this graph here shows the result of a divergence analysis. And what it does is it compares two species. It does pairwise comparisons of species. And so if you think about this, for me, it makes sense. So for vertically inherited biosynthetic genes, so two species, they've been diverging since they split from their common ancestor. And if genes in those species are vertically inherited, then the genes will have been diverging since the species have been diverging. And if housekeeping genes are representative of vertical inheritance, then if vertically inherited mycotoxin biosynthetic genes should reflect, should be similar to housekeeping genes. And so from the trees I showed before, we thought that the humanism gene cluster was vertically inherited in proliferatum and amphiphyllum from a common ancestor, as well as proliferatum and Fusarium oleracum. So, and in both, so proliferatum and amphiphyllum are closely related, so they have a relatively low DS divergence value for the housekeeping genes, and their humanism genes exhibit a relatively low divergence level as well. Proliferatum and oleracum are more distantly related, so their divergence of housekeeping genes is much higher than with proliferatum and amphiphyllum. But also the humanism genes are also exhibit a high degree of divergence. And what you can see here that the humanism genes in this vertical comparison of presumably vertically inherited humanism genes, the divergence of humanism genes is always higher than that of the housekeeping genes, and that's just the way it is. We've been finding that secondary metabolite genes in general diverge more rapidly than housekeeping genes. I'm not sure why that is, but that's how. So if you go over now to the sorry about that. We go over to the horizontal transfer, where we think there was horizontal transfer of the humanism gene cluster. You can look at Fusarium hospi and Fusarium amphiphyllum. They're relatively closely related, and so you see this level of divergence of the housekeeping genes, but much less divergence levels for the humanism genes. Likewise for oxysporum and bovicola. And so this makes sense. So Fusarium hospi and amphiphyllum have been diverging since they split from a common ancestor. So you see this level of divergence, but their humanism genes have only been diverging since the horizontal transfer. So horizontal transfer occurred a long time after divergence of the two species, and we'd expect the divergence of the humanism genes to be less than housekeeping genes. So that's a long-winded way of telling about these divergence values, and I find them very useful, but they're not always 100% accurate, or they can be misleading. So what I try to do is take all these various analyses and do and compare them and look at them. So here's a summary of 4 analyses of 4 gene clusters, monocyte, pulverin, and fuserin, and then something I don't know. I see the gene cluster, but I don't know what the metabolite is. And I can't always tell the specifics. Usually, I can't tell what species were involved in the horizontal transfer, but often I can tell which species complex. So I'm showing you species complexes here rather than species. And so if I look through, you can see whether the data for these examples were consistent or not consistent with horizontal transfer. So for the monocyte cluster, all analyses indicated transfer. The pulverin analysis, the same thing. The fuserin, with fuserin, though, the divergence analysis didn't indicate horizontal transfer, and for the NRPS 11 cluster, only the noting analysis provided evidence for horizontal transfer. So I like to use all these data to make before invoking horizontal transfer, and it's been quite useful. So bottom line is that horizontal transfer contributes to the presence and absence of gene clusters in mycotoxin producing fungi, but you never prove horizontal transfer, but you can have a lot of evidence for or against it. Okay, so now I'm going to switch gears a little bit and talk about, switch gears a lot actually, and talk about production of different analogs of the same type of mycotoxin. And that results from variation in biosynthetic genes. And so our work with the trichothecene mycotoxins and has really provided good evidence for this. So here, trichothecene analogs exhibit immense, tremendous structural diversity. And so here are 5 trichothecene analogs produced by various species of Fusarium. But species in at least 10 other genera of fungi also produce trichothecenes. And so you can see that if you expand out to these other fungi, the variation, structural variation in trichothecenes is tremendous. I think there's like 200 different trichothecene analogs that have been identified. And so when you see this variation, and when I see it, the question is, how does it arise? And we, over the years, our group in Peoria and other groups have identified 21 genes involved in trichothecene biosynthesis, and these genes, variation in these genes can explain a lot of that structural variation. So here's the 21 grid showing the 21 genes and the presence and absence in different species from I think this is 9 or 10 genera, so these genes, most of them encode enzymes, biosynthetic enzymes. Some encode transcription factors, and there's at least one transporter gene here as well. But these genes indicated by the red stars here are ones where they've either been acquired by some species, lost by some species, or their function has changed in some species. And that contributes to the structural variation. So I'm going to just go through a couple of examples here. And so to start with gene acquisition. So most, or maybe half or so of the trichothecenes produced by all the fungi out there that produce trichothecenes, most of those structures lack an oxygen atom at the 8th position. However, 3 genera, Fusarium, Trichothecium, and Mycotoxicosis all produce trichothecenes that can have an oxygen at the 8th position. So that 8th position is shown by the color blue, the orange, and the green. And so evidence for acquisition of genes that cause this or that confer this ability to oxygenate at the 8th position comes from, we see that each of these different fungi, Mycotoxicosis, Trichothecium, and Fusarium, have a different gene that confers that ability to oxygenate the 8th carbon of the trichothecene molecule. So Fusarium uses Tri1 for that, to oxygenate C8. mycotoxicosis uses tri-24, and we don't know yet what the gene is in trichothecium. The trichothecium doesn't have tri-1 and it doesn't have tri-24, so it must have a different gene that infers oxygenation. So there's an example of how fungi have acquired different genes for the same purpose, but the acquisition of genes has changed the structure from trichothecium produced by other fungi. And tri-1 is also interesting in that because it's a great example of change in function, how change in function of a gene can lead to variation in trichothecium structure. So, and we look at so Fusarium has tri-1, and we can see that the function differs in different species. So in Fusarium lanxeptii, Sambucinum, and others, tri-1 encodes a cytochrome 3,450 monooxygenase that catalyzes hydroxylation at the 8th position, whereas in Fusarium asiaticum, some strains of Graminearum and Fusarium poi, and many others, the tri-1 enzyme catalyzes hydroxylation of carbon atom 7 and as well ketylation of carbon atom 8. And in certain strains of Graminearum that produce NX trichothecenes, the tri-1 enzyme catalyzes only hydroxylation at the 7th position. So here's 3 different functions of tri-1, the tri-1 enzyme in different species of fungi. So the function of tri-1 has changed over during the divergence of trichothecium pathways in Fusarium. Okay, and now I don't have it here, but I'm going to show you an example of how gene loss can change structure, contributes to structural variation. So here is another species tree of Fusarium. It includes a lot more species than the previous ones, but again, it shows the different species complexes. And what it shows is that we see trichothecium production in 4 lineages of Fusarium. Sambucinum, Sclerosporium, Inflammatorum equiseti, and then we found one species way down here in the Buharicum clade that produces trichothecenes. And so trichothecene production is limited in Fusarium, although it's widespread in the Sambucinum and Inflammatorum clades. But the purpose of this is to show you that where gene loss can contribute to structural variation. So there's been 3 independent losses of trichothecene for hydroxylation by loss of tri-13. So in Fusarium, for hydroxylation is referred by the tri-13 gene, which encodes another cytochrome 3,450 monooxygenase. And what happens when fungi or species lose tri-13 by deletion or pseudogenation, they lose the ability to put an oxygen on the 4 position shown here in yellow. And so that has happened 3 times independently. The loss of tri-13 has occurred 3 times independently in 3 up here. It's a group of species. Here it's two species, and down here just 1 species. So so you get this structural variation through loss of trichothecene biosynthetic gene. There's another example down here in Buharicum, this undescribed species that we refer to as the yarn species NRRL 6739, member of the Buharicum clade, and it's lost the ability to do the 4 oxygenation, 4 hydroxylation, but it's not because it lost tri-13, because function of tri-13 has changed. And so I'll show you that here. So here is the trichothecene produced by NRRL 66739, simple structure. And then here's the tricluster or triloci that occur in this fungus. And so it's a small tricluster and it has the tri-101 locus with just 1 gene. But you notice it doesn't have the oxygen at the 4 position, but it does have an oxygen or hydroxyl at the 7 position. So if we look at the triloci in other fungi, other Fusarium species that produce, you can see that it's that 66739 cluster is missing multiple genes, and it completely does not have the tri1, tri16 locus. And that's significant because in other fungi that produce trichothecenes with the 7-hydroxyl group, the tri1 gene is which confers that ability. And so the question was how does 6,6,739 produce a 7-hydroxylated trichothecene if it doesn't have a tri1? And so we figured this out by heterologous gene expression. So what Weijia Hao did in our group was she took the tri13 gene from 6,6,739 and expressed it in Fusarium verticillioides, which does not produce trichothecenes. Then what she was able to do, Susan McCormick was able to feed the verticillioides strain expressing the tri13 various trichothecene biosynthetic intermediate to determine what the tri13 does. So here's the experiment. Here's wild type Fusarium verticillioides when it was fed this trichodermin, the biosynthetic trichothecene biosynthetic intermediate trichodermin, which is structure shown here. Fungus couldn't do anything with it. However, when verticillioides was expressing the tri13 from 6,6,739 and it was fed isotrichodermin, it modified it. It put a hydroxyl group on the 7th position, which indicates then that tri13 from 6,6,739 confers the ability to encode the enzyme encoded by that gene catalyzes 7-hydroxylation of humonicins, or sorry, of trichothecenes. So just to put all this information together here, in most species of Fusarium, tri13 enzyme catalyzes 4-hydroxylation, but that function has diverged in 6,6,739, and it now catalyzes 7-hydroxylation. So there's another example of a change in gene function contributing to structural variation of trichothecenes. And then, so the 4-hydroxylation story gets even more complicated if you think about other genera of fungi that produce trichothecenes. So in Fusarium, tri13 in most cases confers 7 or 4-hydroxylation, but in other species of other fungi that produce trichothecenes, they lack tri13, but they have this gene tri22 that confers the same ability. And so based on this, and then the previous slides I showed you with complex changes, losses, and changes in function of tri13, I've come up with a model to explain the evolutionary history of 4-hydroxylation in trichothecenes. So in the ancestral pathway, in this model, the ancestral pathway, the tri22 enzyme catalyze 4-hydroxylation of trichothecenes. So you get a hydroxyl at the 4 position. As time went by, and if some fungi, they retain tri22, and so extant species or extant pathways in mycocyclospora, Stachybotrys, and Trichoderma can produce 4-hydroxylated trichothecenes. And there was a divergence at some point where fungus lost tri22, so it could no longer produce trichothecenes with the 4-hydroxyl or an oxygen at the 4 position. And then during the evolution of trichothecene producing species of Fusarium, they acquired tri13, so that now they could produce trichothecenes with a hydroxyl or an oxygen in general at tri13 at the 4 position. And so if that was retained, when that was retained, then you get fungi like Fusarium sporotropioides that can produce trichothecenes with oxygen at the 4 position. But in some species that you've seen in the previous slide, tri13 was lost, and so the ability to produce 4-hydroxylated trichothecenes was also lost. And then, as I showed you in 6,739, the function of tri13 changed, and the ability to produce the 4 trichothecenes with a hydroxyl at the 4 position was lost, but they acquired hydroxyl at the 7 position. So that's how complicated these scenarios can be with the gain and loss and change in function of biosynthetic genes. So I'm going to. There's another type of type of genetic variation that can give rise to variation in structure, and so that came from work on. Oops, sorry, sorry about that. Okay, and that is what work we've done with my collaborator Santiago Gutierrez on trichoderma. And so, in general, trichoderma species produce simple trichothecenes, and structure two of those is shown here. They're just normal, relatively simple structures of trichothecenes. But there's this group of three species here. I'm going to try to say the names, but they produce macrocyclic structures that have this macrolide ring. And they're called macrocyclic trichothecenes as opposed to the simple trichothecenes. And what's interesting, the only other place where production of macrocyclic trichothecenes is known is with in this clade of species from three different genera: Stachybotrys, Memnomiella, and Myrothecium. And so the question is, how did these three species of trichoderma apparently gain the ability to produce macrocyclic trichothecenes when most other species produce these simple trichothecenes? And we're still figuring this out, but this is what looks like has happened. That is that macrocyclic trichothecene producing species of trichoderma have both trichoderma-like and myrothecium-like trichothecene genes. So here Trichoderma arundinaceum and Brevikompactum have a typical situation of trichothecene biosynthetic locus. They have the tri5 locus, then they have a cluster of 7 genes, and then they have a 3 gene cluster as well. And these loci give rise to the simple trichothecenes shown here. Myrothecium, as an example of a macrocyclic producing fungus, has all its tri-genes in one cluster, and shown here. And what we see with these macrocyclic trichothecene producing species of trichoderma is that they have both trichoderma-like tri-genes loci, and then one myrothecium-like locus of tri-genes. And we're still doing the phylogenetic trees and other phylogenetic analysis to figure all this out, but this looks like to be the case. So what appears to have happened is that this clade of trichothecene producing trichoderma species probably produced simple trichothecenes. It acquired some myrothecium-like genes, and then lost some of its trichoderma-like genes, and that allowed the fungi to now produce macrocyclic trichothecenes. So that, I think this is a fascinating story that we're working on. Hopefully we'll have it figured out in the next few months. But it's another way that variation in trichothecene structure has been generated. Okay, oops. So again, to come back to the diversity of trichothecenes, structural diversity of trichothecenes, there's so much of it. It's just amazing. But it begs the question, why does such variation occur? All these metabolites are toxic. They vary in their toxicity. But why is so much diversity necessary? Structural diversity necessary? I think our work and other people's work on trichothecene production in Fusarium can shed light on this. So in Fusarium head blight of wheat caused by Fusarium graminearum, trichothecene production contributes to the ability of the fungus to spread through wheat heads. So if we knock out trichothecene production, the fungus is limited in its ability to spread within the head. But if it produces trichothecenes, it can infect the whole head. So that's on the fungus side. On the plant side, some plant glucosyltransferases confer resistance to trichothecenes, and that's because attaching a sugar molecule to a trichothecene molecule reduces the toxicity of the trichothecenes. And indeed, some plant glucosyltransferases confer resistance to trichothecenes and provide some level of resistance to fusarium head blight of wheat. So the thing is, though, that these glucosyltransferases can exhibit a lot of substrate specificity. So here is this variation shown in glucosyltransferases from 3 plants: Brachypodium, barley, and rice, and the activity is against 3 trichothecene analogs: deoxynivalenol, nivalenol, and T2 toxin. So if you look at the Brachypodium, it's very effective at attaching a glucose molecule to deoxynivalenol, less so to nivalenol, and not at all to T2 toxin. And you see the opposite occurs with the barley glucosyltransferase. It's most effective at attaching a glucose molecule to T2 toxin, and much less effective at attaching it to deoxynivalenol. And you can see the rice is the same, similar to Brachypodium. So this led me, and I think the others as well, to the hypothesis that the substrate specificity of trichothecene detoxification enzymes drives trichothecene structural diversity. And so I tried to put a little model here. So in the first case, it makes sense to me. If you're a fungus, if there's a fungus that produces a trichothecene structure, and then it's on a plant host, and that host can detoxify that trichothecene, then it behooves the fungus to change the structure so the enzyme no longer recognizes it, or the toxin can avoid the detoxification mechanism. And so that's what I tried to show here. And it certainly would explain the tremendous structural diversity of trichothecenes if fungi are constantly changing the structure of the trichothecene they produce in order to avoid detoxification enzymes. Okay, so now I'm going to switch gears again and talk about non-random loss of genes that results in the truncation of biosynthetic pathways. And again, this work is work I've done in collaboration, and others in Peoria as well. I've done in collaboration with Santiago Gutierrez at the University of León in Spain. And so just to review what goes on in trichoderma. Trichoderma has 3 loci of trichothecene biosynthetic. It has a tri-5 locus. Tri-5 encodes a terpene synthase that converts the primary metabolite farnesyl diphosphate to this terpene called trichodiene. The big tricluster, they call it, has these genes, and most of them encode enzymes that result in a conversion of trichodiene to trichoderma, this compound down here. And then the small tricluster has 3 genes that catalyze the addition of a polypeptide tail to the trichothecene molecule. So that's the typical situation in trichoderma that don't produce microcyclic, that produce simple rather than macrocyclic trichothecenes. So what we see here is that there's a non-random loss of trichothecene genes in trichoderma, and that is evidenced by, and that is loss of everything but the tri-5 gene. And you can see it looks like it's occurred at least 3 times amongst this group of species that have some tri-5 or other tri-loci. And we've seen this with other secondary metabolite genes, other toxin genes, where you see loss of some of the biosynthetic genes, but retention of one or more genes, and it exhibits the same pattern every time the loss occurs. Some genes, specific genes, are retained; the other genes are lost. And so with some work out of Martha Vaughn's lab in Peoria, has provided insight into why that might happen. And so what Martha and Santiago did is they created a strain of the biocontrol agent Trichoderma hartzianum. And just as background, this species of Trichoderma does not have trigenes and it does not produce trichothecenes. So what they did is they introduced into this strain, well, they genetically engineered it by adding the tri-5 gene, the terpene synthase gene to it. So they didn't add the entire trichothecene pathway, they just added the first step in the pathway. So this step catalyzed by the tri-5 enzyme, so that Trichoderma hartzianum could produce this terpene molecule called trichodiene. So the first biosynthetic intermediate in trichothecene biosynthesis. And then what they did then is to look at the effects of trichodiene production by Trichoderma hartzianum on interactions with the Fusarium headblight fungus.
Bob, I think we've lost your sound somehow.
Fusarium graminearum and Trichoderma hartzianum together, and the hartzianum was either didn't produce trichodiene or had the tri-5 gene and produced trichodiene. And what they found is that production of trichodiene by Trichoderma hartzianum inhibited both deoxynivalenol production, the trichothecene production, and inhibited trigene expression in Fusarium graminearum. They also showed that in planta, production of trichodiene enhanced the ability of Trichoderma hartzianum to restrict, or the biocontrol activity of Trichoderma hartzianum against Fusarium graminearum. So the production of trichodiene by Trichoderma hartzianum had a profound effect on the interactions with Fusarium graminearum. And so this led me to the hypothesis that the beneficial activity of mycotoxin biosynthetic intermediates can drive non-random loss of biosynthetic genes. And I thought I'd taken this graph out, so we'll just ignore it. Sorry about that. And so, but if you think about it, if Trichoderma species that have retained tri-5 and the ability to produce trichodiene, that might serve them in the competition with other fungi, including Fusarium graminearum or whatever other fungi they encounter. So, this makes sense to me, this hypothesis, and so it's going to be tested now, but we're thinking about it. So I just like to, I'm getting close to the end here, I just like to address this: how does knowledge of mycotoxin evolution inform control and management efforts? And I think the hypotheses I brought up can help explain this. So in the mycotoxin production versus non-production within a species, it can be used in biocontrol efforts, and there are aflatoxin non-producing strains of the fungus Aspergillus flavus are a very effective biocontrol agent for control of aflatoxin contamination in cotton, in peanuts, and in maize. It's used. The work with cotton is done here in the US and Arizona, and I think Texas as well. But it's being used in some African countries as well. So it's a very effective method to control aflatoxin contamination in some, at least under some circumstances. And then I think the trichothecene structural diversity the hypothesis that trichothecene structural diversity is driven by substrate specificity of detoxification enzyme can help in breeding and engineering efforts to increase trichothecene resistant crops. And then finally, reduction of mycotoxin biosynthetic intermediates non-random loss of genes, could enhance control strategies. And then I think this is where I'm going to show that graph. So here it worked by Martha and Santiago, showing that when they expressed produced engineered trichoderma hartanum to produce trichodine, it resulted in enhanced biocontrol activity of trichoderma hartanum in the wheat head blight system. And so with that, I'd just like to summarize and draw some conclusions. I think hopefully I've shown you today that there's been tremendous variation in mycotoxin production among and within fungal species. And that variation results from several types of genetic changes and genetic, it has several different types of, there are several different genetic bases for that. There's presence and absence of gene clusters and acquisition, loss, and change in function of individual genes within those gene clusters. And we also, so finding out factors that could drive structural variation and mycotoxin production and non-production by contributing to fitness, avoidance of detoxification enzymes, and then beneficial activity of some biosynthetic intermediates. And then finally, I think insights into factors that drive variation in mycotoxin production have the potential to inform mycotoxin control strategies. And then with that, I'd just like to acknowledge a lot of people who worked with me recently. They include scientists at USDA in Peoria, Illinois, particularly acknowledge Susan McCormick and Mark Bussman, who are two chemists. I don't do chemistry, and so I would not be able to do much of what I do without their help. And there's a bunch of technicians in our department. Amy McGovern and Krista Probert work under me, and so they helped me a lot. And then postdocs in our group and the student, I didn't supervise them, but they were instrumental in some of the work that I described today. I have collaborators in other places as well. These include several scientists at the National Research Council in Italy, Santiago Gutierrez and his team at the University of León, and then Teresa Lee at the Rural Development Administration in Korea. And with that, thank you for taking the time to listen.